Skeletal muscles are under voluntary control and are consciously regulated by the nervous system. They are composed of fascicles, which are bundles of muscle fibers enveloped by connective tissue, blood vessels, and nerves. Motor nerve axons supply one or more skeletal muscle fibers, forming a motor unit. Within each muscle fiber, thread-like structures called myofibrils are organized into contractile units known as sarcomeres, which give rise to the striations characteristic of skeletal muscle. Each sarcomere consists of thick and thin myofilaments, strands of proteins known as myosin and actin. Myosin forms thick filaments, while actin forms thin filaments. The protruding heads of myosin filaments create cross bridges that bind to actin filaments in the presence of ATP, an energy transport molecule. Thin filaments consist of a sequence of actin molecules with myosin binding sites. In a resting state, actin molecules bind to tropomyosin and troponin, two contractile proteins that inhibit contraction. The process of skeletal muscle contraction commences at the muscle fiber's plasma membrane, or sarcolemma, which contains invaginations called transverse or T-tubules surrounding each myofibril, connecting to an intricate membranous network known as the sarcoplasmic reticulum. This sarcoplasmic reticulum stores calcium and facilitates the transfer of ions through voltage-gated channels into the myofibril sarcoplasm to regulate muscle contraction. The initiation of muscle contraction involves a series of steps. In the first step, known as excitation, a motor nerve generates an impulse or action potential at the neuromuscular junction, leading to depolarization of the sarcolemma and the generation of an action potential. This action potential spreads through the sarcolemma into the T-tubules, exciting the muscle fiber. The second step, coupling, involves the depolarization of the T-tubules, leading to the release of calcium from channels in the sarcoplasmic reticulum into the sarcoplasm. Troponin binds to calcium and separates from tropomyosin in the third step, known as contraction. Tropomyosin shifts away from actin, exposing the myosin binding sites. This enables myosin to bind to actin, resulting in the activation of ATP and the release of energy, which causes the myosin heads to rotate. As a result, the thin filaments are pulled inward, sliding past the thick filaments and shortening the sarcomere. The final step of muscle contraction is relaxation, where calcium ions return to the sarcoplasmic reticulum, breaking the bonds between myosin and actin. This disengages the cross bridges, allowing the sarcomere to lengthen and resulting in muscle relaxation, bringing the contraction to an end.